Without further ado, I would like to introduce this week's speaker, Dr. Wendy McFadden-Smith. Wendy is the tender fruit and grape integrated pest management specialist. It was a real mouthful, that's why I had to read this, with OMAFRA in Vineland. She has been involved with pest management now for more than 20 years, both in tree fruit and grapes and small fruit. Initially worked for Horticulture Research Institute of Ontario, later University of Guelph, and then moved on to a private consulting company for quite a few years, and at the same time taught here at Brock and was one of the most popular professors here in teaching a mycology course here at Brock, as well as part of a grapevine pest management course. So she still remains an adjunct professor here at Brock University. She also teaches in the Oenology and Viticulture Technician program at Niagara College. So without further ado, I'll introduce Wendy, and the title is New Initiatives in the Management of Grape Sour Rot. Wendy.
Thanks, Andy. Okay, so apparently one of the reasons for my popularity was the jokes I tell about sex, and I've been told I shouldn't do those, so no sex jokes. So may not be as popular as usual. Sour rot is a relative problem for us in Ontario. First really identified and of a major concern about 2005 in Riesling. We started noticing it. And in progressive years, in certain vineyards more than others, we've found that it is a problem. And the one thing that I've learned about sour rot is the more I learn about it, the more I find out there is to learn. So this is what it looks like initially in the vineyard. Can be quite easily mistaken for botrytis. However, with botrytis bunch rot, when you take that berry skin and pull it, the skin will come off and the berry, the flesh of the berry remains intact inside. With sour rot, as soon as you touch that berry, it's juice. So it's very distinctive. The other thing that's very distinctive about it is the odor. Very strong odor of vinegar and nail polish. You can smell it from outside the vineyard in a severely affected vineyard. And this is what a severely infected vineyard looked like. This is a commercial vineyard in 2008 of Riesling. So all of this that you can see here in the clusters, all of this pinky brown coloring is sour rot. The other thing that you'd notice if you looked at these berries is that they will be crawling with fruit fly maggots. So quite a disgusting mess. And this is what you would see on the ground. A lot of crop loss. Growers were requested by the wineries to actually drop crop during 2008 to try and get the level of volatile acidity in the harvested crop down. So what? There is no absolute threshold for volatile acidity. However, most wineries will limit it at about 0.2 to 0.24 grams per liter. It's not necessarily the compounds that are causing the VA that are the issue. It's the organisms that are producing the VA that are the concern. So for winery sanitation, this level is set to try and reduce the amount of contamination by these wild yeasts and bacteria that are not good for fermentation in the winery. In 2008, 20% of the early varieties were rejected at the winery, and this doesn't include the ones that were rejected in the field. Growers were panicking. They had no idea what to do. They were spraying and spraying and spraying, all legally of course, spraying, trying to get rid of this stuff. In addition, every time you have to go through the vineyard and drop crop, there's labor involved. So it was a very expensive proposition. The only real numbers that we have, that should be 2008, not 2009. 2008 crop losses from sour rot or elevated VA through crop insurance with Agricor, about a 1.5 million total crop insurance claim. 750,000 was excess rain, which translated basically to volatile acidity, and then another $250,000 with hail. hail can also cause injuries, which can promote the development of sour rot. So it was very expensive to Ontario growers in 2008. So what's causing it? Sour rot is not unknown in the world. It's been documented in parts of Europe, in France, in Italy, in Germany, in California, but the organisms are different. In 2008, I had an undergrad thesis student, Rhiannon Plant, who did some work looking at trying to identify the organisms that were causing sour rot. So she went in a vineyard that had sour rot in the berries. It was a Riesling vineyard. Collected 4 sets of 20 sour rotted berries, surface sterilized them. So dipped them in ethanol and then passed them through a flame so that all of the organisms that were living on the outside would be eliminated. She then squashed up these berries under sterile conditions and plated out the juice. She used 3 different media, which you don't need to know the names of, but they're supposed to be semi-selective for different organisms. And these were plated at about 25 degrees room temperature for 48 hours. She then did isolations of these, got pure cultures of them, and then we did pathogenicity tests. There's a whole pile of stuff. There are many, many different organisms that were found inside these berries. We didn't know which ones could actually cause the sour rot. So we took berries, grapes, from the grocery store, because this was done in January, surface sterilized them, washed off any pesticide residues, and then inoculated them. So each of these groups represents one type organism. We did this 4 different times in time. The bottom row, the berries were wounded. So we put a scoop of the organism onto the surface of the berry. These top ones just had the organism, no wounding. The bottom ones, we put the organism on and then poked the berry to allow a place for entry to occur. So that's the same throughout all of these. So looking at time course, this would be the first day when it was done. This is day 5. And this is day 8. So you can actually see the progression of organisms. Now this one looks really gross, but it's not typical sour rot. So these were identified as sour rot by smelling and by the nature of the berries, what they looked like. Rhiannon did some ratings, so there was no rot, slight rot, moderate rot, or severe rot, just to give us some relative effect of these different organisms. So the ones that she identified as pathogenic are 1, 2, 3, 4, 5 organisms, and this one was the untreated control, so it wasn't inoculated. The top berries weren't poked, nothing was applied to them. The bottom ones, they were poked, but nothing was applied. So it's just to give us a measurement of background information. So looking at the different isolates, we had 12 that we were looking at. The green bars are the berries that are intact, and I don't know what that is. So the green bars are the ones where the berries were not wounded. The purple bars are the ones where it was wounded. So this is looking at the severity of sour rot development on these inoculated berries. So the ones that we identified as being pathogenic, able to cause the disease, were isolates 3, 4, 5, 7, and 12, just based on the size of the severity that was resulting from that inoculation. Rhiannon went on to use molecular methods to identify these organisms to species, and what she found that was of all of the isolations that she did, 36% were Hanseniospora ovarum, 4% were Candida zemplanina, 49.5 were Gluconobacter cerinus, and 0.3 were Gluconobacter fructuri. So these 2 are yeasts, these 2 are bacteria. So that was the first clue. Granted, this is one vineyard in 1 year. So we realized that this may not be reflective of every vineyard in every year, but it was at least a clue as to develop some methodology and some idea as to how our organisms compare to those elsewhere. Interestingly enough, in BC, similar work is being done. They had one sour rot year, so they got lots of money to do work on it. And what they found was similar species. Hanseniospora and Gluconobacter were the ones that were associated with sour rot in BC as well. So why does it happen? Well, normally where we'll see sour rot is in varieties that have tight clusters or thin skins. The idea being that as the berries start to increase in size, they will squeeze, cause injury, juice will be released, and these organisms will proliferate on the surface of the berries. So the varieties that we primarily see sour rot in would be Pinot Noir, Pinot Gris, Gamay, Chardonnay, Riesling, Gewürztraminer, and Baco Noir. So why does it happen? First of all, think of the development of a grape berry. The amount of wax that's on the surface of a berry by weight is the same when a berry is pea size as when the berry is full size. So that waxy layer becomes much, much thinner. In addition, water will move through the cuticle of the berry. It will move out during the day, the berry will decrease in size, and then at night, when there's dewy conditions, when there's condensation on the surface of the berry, water will move in. The concentration of water inside the berry is lower than outside, so it's going to move from high concentration to low. It's going to swell the berries. So the berries will go through this process of shrinking and swelling and shrinking and swelling, which is fine when it's sunny. On days when it's rainy, that movement of water out of those berries is much less, but the movement of water in is still going to continue. So that berry will continue to swell and eventually will split. Looking at 2008, the weather, everybody that farmed grapes knows it was rainy in 2008. Warm and rainy. Particularly look at September. Between 15 and 20 degrees for the average temperature and elevated rainfall, higher rainfall than average in 2008. Another thing that might have something to do with the development of powdery mildew is a phenomenon called diffuse infections. These are caused by powdery mildew. Now, research has shown that berries are resistant to powdery mildew after 4 to 6 weeks post-bloom. But very close to the end of that time, when they're still just a little bit susceptible, when the great powdery mildew fungus, see these lines here, these are the mycelium of the powdery mildew fungus, it will try to penetrate. So it'll make little tiny wounds in the surface of the berries. So this is looking at, you can't see these diffuse infections except with a hand lens, so you can't tell. On this slide, naked eye, they both look the same. However, when incubated, this is what happened. This is some research that was done in New York by David Godore and his group. So this definitely looks like the sour rot that we see. So powdery mildew is a possibility, may be implicated. Generally, we have always heard, and it is known, that sour rot tends to be associated with bunch rot, Botrytis bunch rot. But in the studies that we've done in the past few years, the correlation isn't really strong. We have frequently found sour rotted berries where there's no botrytis. We found sour rotted berries where there's no berry splitting. So there is a potential for direct infection to occur. Grape berry moth. Anything that's going to cause wounding to the berries. Growers have commented on the fact that if they see a lot of berry moth, they generally get a lot of sour rot. Makes perfect sense. It's causing an entry, a wound site that the organisms can get in. And vinegar flies, or fruit flies. It's been shown that vinegar flies can actually carry, the larvae can actually carry the organisms that cause sour rot in their gut, and as they move from berry to berry in the cluster, they can carry it along and basically inoculate. Plus, when fruit flies land, when adults land on the surface of berries where these organisms grow, and then they move to another berry that's not infected, they will carry the organisms with them. So what can we do about it? Two strategies. The first, reduce the injury. Reduce the ways that those organisms can get in. And secondly, reduce the infection. If the pathogens are there, try and make it so that they can't infect. So the first strategy that we looked at for reducing injury was to look at loosening the grapes. First of all, we're reducing that injury as the berries swell during stage 3. And secondly, there's been a lot of work done showing that where berries are in contact with each other, the cuticle is thinner. So it's more possible for organisms to move from adjacent berries, from one to the next. So in the literature, gibberellic acid is cited as one of the treatments that can be used. Applied at full bloom and 4 days later, it's shown to have fewer berries per cluster and reduce splitting. Andy Reynolds has done some work with this, looking in sovereign carnations for some of the same reasons. One of the reasons that growers are reluctant to try this, or to, well, some of them are, to try this is that there are reports of reduced fruitfulness, especially in Riesling, if you use GA. It's a hormone, so it's going to affect a lot of the different processes that are taking place in the grapevine. When I was in contact with a colleague in Germany who indicated this work that he's been doing on GA, but also that there was another product, product X, which I can't disclose for security reasons. And we decided to try this. The company just isn't ready to release what the product is yet. We looked at it at 180 grams 8 grams active ingredient per hectare applied at full bloom as a way to see about changing cluster architecture. Now in the literature, as I said, loosening clusters isn't new. Zabadal and Dittmer from Michigan reported a study that they developed a rating scale for looking at cluster compactness, where 1 is where the berries are so tightly pressed together that you can't twist the cluster at all. You can't move the cluster. 6, you have most of the berries are not touching anything else, and in between. So we did this treatment and then we rated the clusters for compactness. So remember that a higher number means a looser cluster. So we did this rating at Veraison and again at harvest in both Riesling and in Pinot Noir. And what we found was that this application at bloom, we just did the bloom application, significantly increased the looseness of the cluster. It decreased the number of berries per centimeter rachis, so the rachis was longer, and it decreased the berry weight. This was apparent at Veraison. When we looked at it at harvest, that difference in berry weight wasn't obvious, but in the check, most of the berries had sour rot, so we couldn't really get a good measurement of it. So that one isn't a realistic number. But once again, increased cluster looseness, decreased berries per centimeter. Looking at sour rot, significant decrease in both botrytis, bunch rot, and sour rot when product X was used. Now the effects in Pinot Noir were similar, but we didn't see as much of a difference, so there's no point in showing it as well. We did look at these plots the following spring to determine whether fruitfulness was comparable between vines that had been treated with the product and vines that hadn't, and the number of fruitful shoots per cane was identical. There was no significant difference. So we're good for the following year. Another strategy for changing that canopy architecture is to remove basal leaves at bloom. If you think about the movement of carbohydrate in a grapevine, those basal leaves are very important for providing carbohydrate for fruit set. So if you remove them at bloom just before fruit set occurs, you're starving the clusters. So the idea is that you will have reduced fruit set. Colleagues in Pennsylvania, Brian Head and Jim Travis, looked at this, removing 3 to 4 leaves around vignoles clusters at trace bloom. And what they found was that they had looser clusters with fewer berries. The idea being thicken the wax layer, possibly with increased exposure, as well as loosening the clusters so that you're getting more air movement, the berries don't stay wet as long. Side effects, reduced powdery mildew. If you have good exposure of those clusters, The berries will heat up and possibly be less conducive to powdery mildew infection, and once again reduce botrytis infection. So this is what our bloom removal plot looked like. This is Pinot Noir. This is before leaf removal, and this is after leaf removal. So it's kind of frightening when you do it. It's kind of like when you prune in the spring and you cut off all the canes but two and you think, oh, but we did it. And so you can see that there are no basal leaves. The clusters be in here, this area, so very fully exposed, and the leaves were gone. So in 2009, we included a number of different treatments. We actually had 20 treatments in this trial, including 3 rates of product X, GA at 5 ppm, 10 and 20 ppm. And then we looked at GA at 5 and 10 applied at bloom and 2 weeks later, or 10 days later. We also had a product called Stimplex, which is a seaweed extract that is supposed to have some plant hormones in it that may have had some impact on the architecture of the cluster and our bloom leaf removal. So what you can see from this graph is that product X at 180 grams and GA at 20 ppm had the highest Zabadol reading, meaning they had the loosest clusters. Unfortunately, 2009, what I've found is that when you decide to do a project on something, that's when it's no longer going to be a problem. And 2009 was a good sour rot year for growers, but a really bad sour rot year for researchers. But we can look at incidence. So this is looking at the percentage of clusters that had any kind of sour rot at all. The level of sour rot that we saw was maybe 1-2 berries in a cluster in 2009, as opposed to 25 or 30 or even 50% of a cluster affected. So here you can see that all of the treatments reduced the incidence of sour rot. Here is our product X at 180, and here is our GA at 20. Not significantly different from the check. This one isn't. The rest, all the ones with arrows are significantly different. So they had some effect, but because sour rot wasn't really severe, it was difficult to show these effects. The other thing is that we did samples and ran analyses on all of these plots and tested the adaptability of the analytical services at CCOVI as far as trying to identify VA in samples that had very low levels. And none of the samples that we collected had less than 0.2 grams per liter, so obviously not a big challenge. We also looked at leaf removal by hand at pea-sized berry and Veraison in these plots, and on top of the treatments of product X at 180 and GA at 5 ppm applied twice, we did berry leaf or leaf removal at pea-sized berry. So this is what, at Veraison, this is what the untreated check looked like, where no leaf removal was done. Once again, this is a Pinot Noir plot. We did the same thing in a Riesling plot, so the effects would be very similar. This is leaf removal at bloom, so you can see that even though it was pretty uncomfortable looking at that many leaves being gone, it actually grew quite a few back. This is at pea-sized leaf removal, so there is pretty good exposure of the clusters. And this is the Veraison leaf removal. Pretty stark. A lot of vineyards in Niagara in 2009 looked like this because growers were so desperate to try and get their fruit to mature. Looking at cluster weight with the Riesling, you can see that all of the treatments reduced cluster weight. Pea size was the only one that was significantly different from the check, where there was none. The big effect was in the Pinot Noir. That bloom leaf removal actually significantly reduced the cluster weight. So it may have some applicability as far as looking at it for a cluster architecture manipulation. Soluble solids with the Pinot Noir. significant reduction in soluble solids with the leaf removal. Those vines needed those leaves last year. Taking them off to increase exposure kind of worked against what they were there for, which is to fill the grapes with sugar. And the effect on the Riesling was much less notable. So we're going to play around with this again in 2010. Hopefully it'll be a drier season with lots of rain at the end for sour rot. Just in my plots. Just in my plots. How's that? Nowhere else but in my plots. We did look at leaf removal with calcium as well. The idea being that calcium may either affect the ability of the cells to withstand fracture when splitting was occurring, or changing the osmotic potential outside of the berry so that inside and outside the berry, when there's water, would be relatively similar, so the movement of water into the berry causing the swelling would be less. And we did see a reduction in the incidence of sour rot when calcium was applied either twice or 4 times. Now we did see a bit of burning when we applied the calcium 4 times. I think that was a rate issue. As far as sour rot in the Pinot Noir, once again it was brought in during a dry period in September, one of the few dry periods that we had, and there was a lot less sour rot in it, so I couldn't show these differences. So another strategy reducing injury, took a look at some literature from cherries, because in Niagara the 2 times when you can count on rain happening if you're a farmer are at cherry harvest and a grape harvest. Splitting in cherries is a big problem. There's been a lot of work done on trying to ameliorate cherry cracking. So just some of the strategies: physically removing it, so blowing it off with helicopters or air blast sprayers. We didn't try that one. The osmoticum sprays, the calcium chloride treatment, we tried that one. There are products such as Rain Guard, which is a surfactant, which theoretically will cause the water to run off of the berries instead of staying on the surface. So I've been in touch with Rain Guard about getting some of their product. Plant hormones, copper, surfactants. Just as a summary, very, very variable results. And then calcium to strengthen the cell walls. The timing is suggested as fruit set to Veraison. So in 2008, we looked at applying calcium at cluster closure, Veraison, and 2 weeks post-Veraison. We did oligosol calcium at 10 liters per hectare. We also looked at a product called Acadian kelp, which is a seaweed product, and our standard was a Scala Elevate rotation. Looking at botrytis, this one really surprised me. I didn't think it would happen, but it did. This is when the null hypothesis is actually accepted. All of the treatments significantly reduced botrytis, and all of the treatments significantly reduced sour rot. Interestingly, the fungicide treatments didn't do much as far as controlling sour rot, which makes sense because the organisms that are associated with it are not ones that are susceptible to fungicides. We also did it in Pinot Noir and Riesling, applying it at pea size, Veraison, or at both times to see if it made a difference. Whoops. And pretty much the same type of response. There was a reduction in sour rot using the calcium spray twice, more than just once or the pea size. So once again, calcium may have some fit. In 2009, we switched products to Stop It, which is the only one that has grapes on the label, applied 2 or 4 plus pea-size leaf removal, and I already showed you those, and it did reduce the amount of sour rot. Potassium metabisulfite is something else that we've been tossing around looking at as a possibility. It's used as an antioxidant and antimicrobial in the vineyard, or in the winery. Fairly low rates though, 40 to 60 grams per ton of grapes. When we first started seeing sour rot, I did a bit of a search on the internet and found work done in New Zealand and Australia looking at using potassium metabisulfite, KMS, for botrytis management. Gave pretty inconsistent results for botrytis. However, it did give me an idea for a rate, because no phytotoxicity was reported at up to 5 kilos per 1,000 liters. So that's what we decided to try as our rate. So the questions, first of all, does it do anything? Does it work? How does it work? Is it an antioxidant? Is it an antimicrobial? Does it dry up the berries? What does it do? Is there a problem with it? Will we have excess sulfites in the wine if we apply it? Because sour rot will generally occur in the last week before harvest, and that's when you need to get in to try to control it. Work and equipment exposure is really important. We didn't address this, but it's a very strong oxidant, so you have to be very careful when you're using it, especially mixing it, to take proper precautions. So in 2008, we went into a Riesling vineyard that had a history of sour rot. This is the one that we had done previous work on. I went through and removed all the clusters that had more than 25% sour rot in the cluster. So some were 100, some were 50, nothing above 25 stayed. It all went on the ground. This is a pretty strong test for KMS, by the way. 25% is well above what you would reject in a winery. So we sprayed it on day 1, collected 25 clusters per plot, and then determined the VA on those clusters. And this is what we found. The other treatments that we used were chlorine dioxide, which is a sterilant used to sterilize water. Pristine is a fungicide that has sour rot on its label. Copper. Millstop is potassium bicarbonate, another one that growers have tried to look at for reducing sour rot, and then the KMS. And what we found is that the Millstop, all of the treatments reduced the amount of sour rot, but the Millstop and the KMS did the best, and they were significantly less than everything else. So this is still well above the 0.2 to 0.24 grams per liter that would be acceptable in a winery, but very, very challenging for the treatments. So if we were in a commercial situation with the level of sour rot that we'd normally see, hopefully it would bring it down more. In 2009, Christina Huber started working with me doing her graduate work, and one of the questions we had was, when is the best time? When do we need to spray for sour rot? When do we start? Do we start at Veraison? Do we start 2 weeks later, etc.? So we developed an experiment, which took a lot of work, where one treatment received KMS treatments 6 times, starting at Veraison, 2-week intervals for the first 4, and then these 2 were at 1 week, 10-day intervals. Started the next one 2 weeks after Veraison, all the way through, 4 weeks after Veraison, all the way through 5, 6, and this one was sprayed the day before harvest. So this was supposed to give us an idea of when we needed to start. The other thing that we looked at a variety of, this was a shotgun approach. We looked at a variety of different compounds to see if any of them would have an effect. So we looked at KMS, that 5, this is not ppm, it should be, should be grams per liter. I get it wrong every time. There should be 5 grams per liter per 1,000 liters, 5 kilos per 1,000 liters, 10 kilos per 1,000 liters, 2.5 kilos per 1,000 liters. So please don't look at the ppm because that's wrong. We're trying to determine the rate. Millstop plus KMS, oxidate, which is a peroxide product. These are applied at the same interval as that full spray interval that I showed you in the previous slide. Actinovate is a biocontrol product that contains streptomyces. It has sour rot on the label in the States. Blight-Ban A506 is now registered in Canada. Actinovate is as well. Blight-Ban A506 has sour rot on its label in the States. And Pure Shade is a product that, it's a calcium carbonate that is registered as a sunscreen, a sun protectant, but there was some observations that there was less sour rot in vines that received that treatment as opposed to vines that didn't, so we figured we'd throw that in. And then we had 3 applications at Veraison, 2 weeks post-Veraison and 4 weeks post-Veraison. of vermicompost. It's a tea from worm compost. Switch, which is a fungicide that's registered in the States and has sour rot on it. And somebody had heard that it worked on sour rot. And Stop It, calcium chloride, and then we had an untreated check. And then we had no sour rot. We had no phyto either. But, so just, why didn't we have sour rot last year? Aside from to make Christina's life more challenging. This graph shows the precipitation and temperatures from May through October in 2008 in blue and 2009 in green. And what you'll see is temperature-wise, looks pretty similar. 2009 felt cooler to me than 2008, but not according to the loggers. Rainfall-wise, we had more rain in 2008 than 2009. This is the critical part, from September to October. Oh, sorry. First of all, this is when Pinot Noir harvest occurred in 2008 and Riesling, and this is when it occurred in 2009, so about 10 days late. So take the entire development of those berries and shift it 10 days later. It's going to be cooler 10 days later. So if we look at September of 2008 and 2009, what you'll see is September was actually pretty dry in 2009. Whereas in 2008 it was quite wet. We had one little blip of rain close to harvest, which is normal for grapes, but the temperature really got cool as well. So these are organisms that are going to function, they're going to reproduce more at temperatures 20 to 25. The other thing that was really, really notable in 2009, no fruit flies. No fruit flies. Not a maggot to be found. So that vector was not there. This is some work some data that Christina brought together. We looked at Brix development and sour rot development, wondering if there's some relationship, whether when the Brix get to a certain point, those organisms are more able to multiply. The only thing that we found was when it warmed up and it rained, sour rot increased. So looking at post-Veraison treatments with berry microflora, Christina sampled fruit before and after these treatments to see what effects these treatments would have on the organisms that are present on the surface of the berries. So these are the treatments that she sampled, squished them, plated them out. These were not surface sterilized because she wanted to know what was on the surface of the berries. And then she counted all of the colonies on all of these plates. She had no wrinkles when she started, and now she has squint lines from looking at these plates for hours and hours and hours. This is a summary of just some of the information that Christina has found, that with the KMS, all of the treatments reduced, this is just yeast, the percentage of yeasts that were present on the surface, which is kind of interesting when you think of vermicompost, because it would have had all kinds of stuff in it. Millstop seemed to reduce the yeast population the most, and this kind of coincides with what has been found in BC, where in vitro tests they have been exposing the different organisms, and they found millstop tended to knock the yeast down the most. The last question is, what does KMS do to the wine? So we looked at treating vines 2 weeks, 1 week, 3 days, and 1 day before harvest with the 5 kilograms per 1,000 liters, which works out to 5,000 ppm, not 5. And this is a rate of about 2.5 kilos of KMS per hectare. And this is applied only in the fruiting zone. For all of these treatments, it was applied only in the fruiting zone. These were all Locane VSP, vertical shoot position plots. So each plot consisted of rot-free. We didn't want to look at the effects of the sour rot. We just wanted to look at the effects of the KMS on vinification. So we took healthy berries and made wine out of them. So the fermentations were sampled every other day. The number of cells were counted. The Brix was determined until these wines were fermented to dryness. Looking at the fermentation curve, basically no significant difference. The only thing that's kind of interesting is this is the one that received no KMS. So fermentation was actually a little bit later, a little bit slower. This is cell count, looking at cell numbers, no effect. I know this doesn't look like a normal fermentation graph. There's a story behind that. But so the KMS residues were not affecting the activity of the yeasts. And looking at quality parameters, no significant difference in TA, residual sugar, or ethanol. So no difference among those different treatments as far as vinification is concerned. So even at 1 day pre-harvest, the residues were not infecting vinification. We don't know what the effect is on the storability of the wine. This is a fourth-year thesis project with a limited time to complete the study, but it's something that we might want to look at. Canopy management is something that may affect the development of phenolics, wax deposition, and Dr. Ingalls, Dr. Helen Fisher with the University of Guelph and I have a large project that we're working on, and one of the things that Dr. Fisher will be looking at is the effect of the timing of leaf removal on some of these characteristics of the berries. So we'll be looking at return fruitfulness. We'll be continuing to look at these cluster loosening treatments. In some in vitro studies, we'll be looking at the organisms that are identified as pathogenic in Christina's thesis work and looking at them at different temperatures and different relative humidities and different wetness durations. The timing of the treatments, hopefully we'll have pressure somewhere that we can look at when we actually need to start treating. I have a number of new post-Veraison treatments. One of them will be looking at ozone as a sterilant of the berries to see if it will help knock down these populations. Interactions among the causal organisms with botrytis and powdery mildew, and as I said, effects of the treatment on cuticle and skin characteristics. So it's a big project, and there are a lot of people involved. The Ontario Grape and Wine Research Inc. and the Niagara Peninsula Fruit and Vegetable Growers Association have funded us significantly. Vincor has been very supportive in donating my vineyard, my sour rot heaven, for me to work at. The only challenge being chased through the vineyard once in a while when the sprayer needs to get through. Schenck Greenhouses and Farms Limited also donated a vineyard for us to work in, and we've done some work with Niagara Vintage Harvesters as well. Whoops, wrong way. A lot of people involved. Debbie Ingalls is the one who handles all of the winemaking stuff. Lisa Dowling and Rhiannon Plant, their fourth-year theses. Christina Hubert, the grad student involved. Catherine Hoshku-Toombs has given us technical assistance. Postdocs and graduate students in Debbie's lab. Colleagues in Ag Canada at Summerland have helped us with some of the molecular techniques, and Keith Seaford at Ag Canada helped identify some of the molecular organisms, some of the mycelial organisms. And finally, the chemical industry has also been very gracious in donating product, and so I'd like to recognize all of them for their help as well. Thank you. Are there any questions? I've said it all. I wore you out?
What about for this year?
What about for this year? You're buying lottery tickets, Ken. I'm going to ask you. The question is, any predictions for this year?
We're 18 days ahead of normal.
18 days ahead of normal. For fruit trees, Baco is pushing somewhere in Niagara-on-the-Lake, I've heard. It's difficult to tell. The Farmer's Almanac says cool and wet again all summer. That's about as good as I can get. Ed?
What would be your feeling on cutting off at pea size, the bottom maybe 10 or 15 berries per bunch, so that you get more air that travels up into the bunch, or it's looser when it starts to swell a little bit?
The question is, what about removing the bottom 15 to 20 berries at the bottom of the cluster around pea size. Head and Travis actually did some of that in their research. They were looking at vignoles, and their idea was that if you remove those bottom berries, the water would drain out better. And they did find a reduction. It's just incredibly tedious to go through and with scissors cut the bottom of every cluster off.
Well, if you count the clusters anyway, you might as well cut off the bottom.
If you're counting clusters anyway and you want to cut off the bottom, then you can try it. But it has been looked at somewhere else. I was hoping that there'd be a way to mechanically do that, but I haven't figured it out yet.
What about leaf pulling? What if it's running mechanical? Does it cost instead of hand? So, you're pulling leaves, trying to get the base leaf all the time with it.
The question being, is mechanical leaf pulling adaptable to this? I don't know if I would go with the basal leaf pulling because your chances of mechanically removing the leaves at bloom, because your chances of injuring the clusters are very high. I would say that mechanical leaf pulling at fruit set to pea size is probably going to help. It'll help with botrytis for sure. It's been shown to help with powdery mildew, and can't hurt. I'm not comfortable with the big leaf stripping at Veraison.
What was the rates of the millstop and the KMS? Just thinking one's a base and one's an acid.
What were the rates? The KMS was 5
grams per liter. No, the millstop is 12 grams per liter.
So 12 kilos per 1,000 liters of millstop, which is label, and 5 kilos of KMS.
12 millstop, sorry. 12 millstop, 5 cans. And we ended up reducing this.
And we couldn't tell what the effect was when you were mixing them this year, but last year it seemed to be synergistic. So I don't think there's a canceling out effect. And there was no phyto.
Wendy, in your data, I don't think you included a graph that just showed residual levels of sulfur dioxide in the fruit even after 1 day of KMS spray. Can you just comment that those levels were really low and at detectable levels of measuring SO2?
Throughout all of the trials in 2009, the KMS levels were below whatever we could detect, which is why it was so challenging for analytical services for the most of the treatments, even with 1 day pre-harvest.
What is your gut feeling about the diffuse powdery mildew infections causing damage? What's your gut feeling about how big a problem that actually is in your salad?
The question is, what's the implication of the diffuse powdery mildew infections? We've shown that some of these organisms can infect directly on healthy fruit, but those diffuse infections may pump it up. 2008 was very wet. It was a good powdery year for a pathologist, bad for growers. There was powdery mildew showing up in 2009 as well, but not to the same degree. So it may be that we need to just make sure that we have those fruit covered a little bit longer. We've been focusing from bloom to 4 to 6 weeks, which is pretty close to Veraison for our treatments. Once you get past that, they're not going to be susceptible anymore. Okay, thank you very much.